So, dear participants, I once again welcome you to the second session of today's schedule. So, we have Dr. Ritura Purohit with us. He will be giving you an insight to molecular dynamics simulation. The title of his talk is Computational Intervention in the Identification of Lead Molecules. He is a principal scientist in Department of Biotechnology at CSIR Institute of Himalayan Bioresource Technology, Palampur, Himachal Pradesh. He is our notable alumni. He did his master's in technology from Indian Institute of Information Technology, Allahabad. Thereafter, his PhD from Vellore Institute of Technology, Vellore, India. Then he went for his postdoc to Italian Institute for Genomic Medicine, Torino, Italy. And thereafter he joined School of Biosciences and Technology, Vellore Institute of Technology, Vellore, India. And from 2014 onwards, he has been a member of CSIR Institute of Himalayan Bioresource Technology. He has more than 92 publications to his credit. His H index is 27 and he has several key publications to his credit. He is currently PI of many projects, many CSIR projects, and is holding key positions at CSIR IHBT. So now I welcome Dr. Rituraj Purohit on behalf of the organizing committee and all the participants. So it's over to Dr. Rituraj Purohit.
Thank you, ma'am, for introducing me. So last from 2 days, I'm listening also this all the lectures from Dr. Mishra to Dr. Katara, and you. They all gave very nice talk about the computational approach, different computational approaches, and I hope you are getting benefit of all this discussion talk given by all important people. So today I'm going to cover MD simulation through my talk, and in this I just want to make you aware about how why MD simulation is required and what kind of properties, what kind of output we can observe in this simulation, and why we do this type of simulation, and if the simulation, if the MD simulation is not there, how we can get the particular property of the system, is it possible to get or not, and how it is laborious. Everything we are going to discuss in this talk. So as you know that lead molecules is nothing but the hit molecule or the important molecule which is there in the pool and have biological activity or pharmacological activity. So to identify the particular lead molecule, we have some computational techniques, and through that technique we can easily observe, or you can find out, you can easily screen those molecules, and later on you can check the efficacy or real pharmacological activity through experimental techniques. As we know that the biological system, I mean, we have the lot of protein in our body, and in general that protein have 20 amino acids, that is in the form of sequence, which is there in the, generally we used to retrieve from the NCBI database. So it is a notion that the sequence, if you have the sequence, you can predict the structure, and if you have the structure, you can predict the function. So ultimate task is to get the function of particular protein or the sequence which we have. So as you know, there are many sequences given in the database, but we do not know the structure of all those sequences because we do not have the enormous capability like X-ray or NMR. We have the X-ray and NMR, but to process each sequence or to process each structure, to get the structure, we need to do a lot of work. And it's a very hard work behind to elucidate a single structure. And with the homology, if you see the sequence, the many sequence can give same structure. The side change pattern more important than the sequence. So it is. You already saw in the previous lecture also that the sequence is quite a it's just a string in the text form. AGCT. If you talk about the protein sequence, but if you go for the structure, I mean, if you are modeling the structure and if you are trying to get the structure of a particular protein, then the structure is very specific because it is just directly connecting to the toward the function of that particular protein, and the you can say the orientation of each amino acid in the three-dimension space is, you can say that is the structure of that particular protein. So when we see the homology is more than 50%, like to have some same structure and function, it is I mean, it is a notion that if you have the homology of 50% or more than 50%, then you will get the similar kind of structure. But there may be I mean the contradictory here also. So, and the other thing is core conserved and surface and loop variables. So 3D shape more conserved than the sequence. That's why we always bother about the 3D structure, or you can say the structure of the protein, because even though the sequence are same, but if there is a minor change in the sequence, you will get the different function. There are limited number of structure framework, as I told before, that we have very limited number of structures compared to the sequences. How we get the three-dimensional structure of protein? So there are some procedure, experimental techniques. As we know that we have the two major experimental technique, that is X-ray and the NMR. The X-ray will through X-ray we will get the stable fold, good quality crystal. And NMR, again, this is I mean, there is a slight difference in that. Some protein which is not soluble will go for the NMR, and it's not crystallized. Sorry, not if you are not able to get crystal structure, we'll solubilize it in particular solvent and we'll get the NMR. So both the technique are very robust, and through these techniques, we used to find the three-dimension coordinates of particular protein or DNA or macromolecule. On other hand, we have some in silico method. Through that, we can get the structure. Sequence or profile alignment, use a similar sequence. I mean, the same way. After getting the structure of through experimental, we had to do some kind of computational. We had to implement some kind of computational approach also. But if you do not, if you cannot perform X-ray or NMR, we will have some kind of computational techniques, and through that you can get the structure. So that is protein sequence or profile alignment, threading, and amino acid structure prediction. So I'm not going to going in depth in that because my I need to cover the MD simulation. I'm just giving the brief of all these techniques to start. Okay, so we have computational method for protein structure prediction. First is homology and comparative modeling. In that, fold recognition, threading approach, evaluation method that utilize knowledge-based information, and evaluation method without the knowledge-based. So homology modeling, generally in this kind of technique, we have the backend of the modeling is the structured database. You should have the structured database, so that you can always refer your carry sequence. You can compare your carry sequence with the structure database, and if similarity is there, you can get the particular structure as a template. Why do we need computational approach? I mean, what is the need? I mean, it is not always, I mean, if you have the experimental techniques through which we are getting correct or exact or real-time structure, why we need it? Why we need other type of approach, like, or you can say the computational approach? So the goal of research in the area of structural genomics is to provide the means to characterize and identify the large number of protein sequences that are being discovered. As I told, there is a big gap in between that whatever the sequence we have available sequence in the database and the structure. The structure database is so small compared to the sequence database. You can see in the NCBI database you have huge sequences of different kind of protein of all organism, but you have very limited structure compared to that. So knowledge of the three-dimensional structure is very important. As in the first slide I said, if you have the three-dimensional structure, you can predict the function. And if you know the function, you can say about the activity of the protein, you can modulate the activity of protein, you can observe actual activity of that particular protein. You can add up, or you can minimize the activity of that protein. For that, you need to have three-dimensional structure. Like if you want to perform docking, as you saw in previous lecture, that for that also you need starting structure, right? Three-dimensional structure you need. Helps with the rational design, as I told that you for rational designing a drug design, you need to have three-dimensional structure of the site, or other aspect, the site-directed mutation. As you know that we have a lot of sometimes, I mean, through environment or kind of chemicals, we used to get mutations. Like or other example, you see in the SARS virus, the virus is getting a lot of mutation. Why? Because the virus want to survive, or want to live prolong in your body or in the environment. So that's why the virus is getting mutant. So through mutation, virus is changing their key, the three-dimensional structure of key enzyme, so that they can perform. I mean, the virus can perform their work, but the killing agent or inhibitors could not kill could not kill the virus. So that's why, if you want to study the mutation, what kind of mutation is important for the virus, and what kind of structural changes occurring through that particular mutation, everything you need to have three-dimensional structure. Can be a great importance for the design of drug. As I told, for drug designing, you should have the structure, and the structure quality is also important for the drug designing. If you have this, because the drug designing, you need to start from structure, and if you have very good structure, you can really determine the actual pose of the lead candidate, and through that, what are the bonding occurring between the lead candidate and the protein, or you can say the structure of the interest. Greatly enhance our understanding of how protein function and how they interact with each other. For example, explain antigen behavior, DNA binding specificity. So for all these, we need to have protein structure. If you want to observe some kind of interaction between two important protein like antigen antibody, then you should have the structure of those antibody and antigen. With a better computational method, this can be done extremely fast. That's why we need computational approaches. Because the X-ray and NMR or cryo or all these techniques are very laborious, time consuming, a lot of, it's very, I mean, you have to put a lot of money in that, because to install it and to create a facility of all this, we need to put a lot of money in that. But on the other hand, if you have the computational technique, you can get the structure. Nowadays, it is possible to get about 90% on with the help of computational approaches. Like first approach, which was homology modeling, which was based on following steps. You can see the template recognition, alignment, determine the structurally conserved region, backbone generation, building loop, confirmation search, refinement, and the validation. So the very first step is template recognition. Is for this also you need the structured database. Like homology modeling process is basically based on the structured database. You should have the proper structured database. Through that, you can search the most similar, or you can say the identical sequence from the structure database that will act as a template, and the template is nothing but your you can say the original. I mean, that will be the use. That is only you can say that if it is 90% similar, the template is perfectly fine compared to the 50% similarity. So that template will provide you the further by adding the loops and the backbone, you can complete the structure. So the template is to search a template, it is very important to check the identity of the query sequence and the database sequence or the template sequence. Then after that, you have to align them, and what are the lagging in between, where is the coverage, what are the coverage, and which portion is not covering, that you have to go for the loop modeling, backbone generation, and all this technique. And after that, you need to validate it. Either this is the structure which you got from homology modeling, or fulfilling the criteria like Ramachandran plot, energy, and everything you have to check. These are some of the automated web-based homology tools which generally we use for homology modeling, SIS model, WhatIf, 3D Jigsaw, hybrid 3D, and we have some evolution for model evolution. We have some another few servers which is mentioned here, and a standalone software also we have for modeling protein structure, like Composer Modeler is very famous in this area. Insight 2, Soluble. What are the challenges? I mean, we have the structure, we have the structure from coming out from the computational technique or homology modeling. So what are the challenges? I mean, to model a protein with lower similarity is the main challenge. I mean, if you have the similarity of your query sequence and the database sequence lesser than 30%, then you are in trouble. So you cannot predict your structure through homology. To increase accuracy, model and to make it fully automated. Again, it is not automated. You have to check, you have to select template based on your consciousness, and after that, you need to finalize the loops, which particular sidechain is important, either it is lying in the conserved domain or not, everything you have to check. So it is not automated. To make it automated, it is a challenge. So improvement may include simultaneous optimization technique in sidechain modeling and the loop modeling. Again, if you got the 90% the structure of your protein, desired protein, but if you are not getting the key loops, if you are not finalizing the key loop, then again at that point also you are losing the accuracy. So this is also one of the biggest challenge in the homology modeling, to optimize a loop, which sidechain is appropriate, because we have the vibration and rotational parameter and the degree of freedom, different degree of freedom for each atom, and to finalize which particular degree of freedom is appropriate for the structure or actual with the like in the nature, it is very tough. Developing better optimizer and potential function which can lead to the model structure away from the template. So again, we have for this also we have some potential function and optimizer also we have to optimize the loop and modeling. But this is one of the big challenge in this area. Okay, so after all this, you might have already done it, homology modeling and all, but this is a kind of very new, recently launched. I think near about 4 or 5 months ago, you heard about AlphaFold. This is another very efficient technique based on the AI developed by the Google DeepMind group, which perform the prediction of protein structure. The program is designed as a deep learning system. So again, for this also you need to have the structured database in your backend. But here they implement deep learning, artificial intelligence concept to get the structure. And for this, AlphaFold finds similar sequences to the input, extract the information using the spatial neural network architecture, and then pass this information to another neural network that produce a structure. After generating a final structure, it will take all the information and pass it back to the beginning of the evoformer block. So they have two blocks, evoformer blocks. Through that, this DeepMind are AI blocks, you can say, and through where the particular process or align the structure set as well as the sequence set. Here you can see the architecture, which is recently published few months back. In this, the user will put input, and the input goes to the genetic database search, and the other at the same time structured database search. Both the through genetic database search, you will have the MSA and the structured database. It finds template, appropriate template. Again, it goes to the evoformer block, and this evoformer block process it, and another evoformer block is there to which check the structure, and finally we'll have the three-dimensional structure of the protein. So recently the AlphaFold predict all kind of protein present in the human body. You can imagine we have a lot of protein in our body which is unsolved. We do not have crystal structure of all the protein, but the first task this AlphaFold done that they model all kind of protein. It is not available currently, but they claim that they model all kind of all proteins of human body, and within I think 24 hours or 2 days or something like that, very short time, and the accuracy is very high compared to the other server or software which we have all like iTaser and all. The accuracy of this is very high. Then in the caps, it is on the top. The AlphaFold is on the top. So after having the protein structure, either it is from experiment or computationally homology modeling or whatever, we need to for lead identification, we need to have another counterpart that is ligand. So ligand is a molecule substance that forms a complex with the biomolecules to serve the biological process. So lead molecule is the chemical molecule, right? It's a chemical entity, but all the chemical molecule cannot be lead. That is the main difference between the chemical molecule and the lead molecule. So lead molecules are able to perform biological or pharmacological function. So we have the source to extract the lead molecule. It is from Ligand database, Zinc, DrugBank, PubChem, or if you have the in-house library, chemistry department, that they are generating a lot of molecule in their lab. So we have the library in that, so one can retrieve the chemical molecule from in-house library or co-crystallized structure. And after getting it, you need to optimize it. As you saw in the last lecture, you need to optimize the ligand molecule, add appropriate charges, and minimize the energy. Right? If you are not minimizing or optimizing your ligand molecules and directly implementing for docking, then you will have the whatever the score or energy you are getting, that will not be actual one, right? So always you need to optimize it before implementing for, or before using in the docking experiment. So after having protein and the ligand, you need to go for some kind of techniques. Through that, you can join these two molecules, right? So we have molecular dynamic docking procedure, as you saw elaborately how we can do it. What is the concept? What are the things to perform everything? Already Dr. Pramod had explained. So new drug very well complemented with molecular docking techniques. I mean, if the chemical molecule are able to fit into the cavity of receptor and are able to perform or biological task. So you cannot observe biological task in the computer, right? So you can only observe the bonding behavior. So suppose one example I will give here. If you have a drug complex, suppose aspirin complex with a particular receptor, right? And if you want to search a chemical molecules like aspirin, right, which has an aspirin-like activity computationally. So how you can see that if you have the crystal structure of this particular molecule, that will be the reference. Remove the aspirin from the cavity, screen your molecule, all the molecule, whatever molecule you have with this receptor, and try to find out which molecule has the best affinity first. And second, check the bonding behavior. If the molecule are able to make all kind of bonds like aspirin is making with the receptor, then this molecule could perform same biological action like aspirin. So in this way, you can observe the biological, you can relate the biological or pharmacological action, right? So bonding behavior is very important, like aspirin 25 present in the protease, HIV protease, and it is making bond with the sequenavir, right? So and it is perfectly fit into the cavity. If some other molecule are able to make a bond with aspirin 25, aspartic acid 25, and having good affinity with the receptor, then this could be a potential lead, right? So another goal of molecule docking is to rank the ligand. We have a lot of chemical molecule, and if you want to rank it, you can use this docking score. And several online servers and paid softwares are also available to perform the docking. So this is a very general pictorial representation of docking, where we have the receptor. This is the Pcna of human and the ligand molecule. So in this case, we have the crystal structure. In the table, you can see that we have the standard inhibitor, or you can see the co-crystallized inhibitor, and the two other bioactive molecules. This is hypersine and pseudohypersine. These two molecules are there. It binds, and it is in the right side. You can see that the co-crystallized ligand on the top, and the other green color bioactive molecules are dog in the PCN, and you have the binding energy here. So best binding energy is pseudohyphracin. So this is one of the what is the bioactive which can perform similar biochemical, or you can say biopharmacological action like standard molecule. So in this case, you can see the bonding behavior like histidine 44 blue is making bond with the standard inhibitor, right? And the same way you can find the hypersine and pseudohypersine is also making the bond with the histidine 44. So if you read the crystal structure paper, you will get the information that histidine 44 is very important. Likewise, other residue is also there in this. That is 131, I think, 126, 129. Okay, but only the histidine 44 is making. So why this picture is important? Because in this paper it is given that the histidine 44 is very important for biological action. I mean, for like if a ligand is making a hydrogen bond with the histidine 44, are able to retain on the binding side, as well as it will have the biological action. So based on this, you can rank the bioactive molecules, and we ranked it, and we got the pseudohypersine on the top. Now, after having this complex means the protein ligand complex. From docking, we have some score, right? But as you know that the docking will only provide the static pose of ligand, or you can see the protein and ligand static pose. You do not have the dynamic pose. What will happen after 10 seconds, you do not know. Either you will get the ligand on the same position or not, you don't know. So for that thing, you need a protein dynamic simulation. Right, this complex should be simulated at particular time, for particular condition, physiological condition, and during the simulation, if the complex is retained, you can say or the ligand is retained in the cavity, then you can say that protein ligand complex is, or you can say that lead or that molecule is have a drug-like activity. So with the advent of computers, Like why I mean the molecular dynamics is required. So before that, you need to know that where we can do that, right, in the computer. So with the advent of computers, we all chemists, physicists, material scientists have begun to exploit the power of computers for probing the property of material through simulation. So we need to probe the properties of materials, whatever the materials in the form of complex, protein ligand complex, nuclear material, or biomolecules, or life processes. All this material, if you are able to probe the property, we need to have a proper computer, and we need to know the molecular dynamics simulation kind of approach to probe the properties. Almost all kind of material or physical world can be probed for their properties by designing appropriate simulation algorithm. So molecular dynamics, where the constituent of the system are allowed to interact according to known law of physics. So at that time also, I mean, if you what you say, if you performing molecular dynamics, you should with the help of computers, you are applying the law of physics over a particular time of period on the system, right? So that's why we need a molecular dynamics simulation. So for before starting, I will give some points for the introducing the molecular dynamics system. I mean, what is the MD simulation? It is a computational method that simulate the motion of systems or particles. The goal of molecular dynamics simulation is to predict the macroscopic properties such as pressure, energy, heat, capacity, and etc. For the microscopic property, including atom position and velocity generated by molecular dynamics molecular dynamics simulation. And Maclaurin introduced the first protein simulation in 1977, and since then this method has been widely used in the theoretical study of biological molecules, including protein and nucleic acid. A molecular dynamic simulation generates a sequence of points in multidimensional space as a function of time, where the points belongs to the same collection of all possible systems which have different mechanical state. The ergodic hypothesis, which states that the time average equals the ensemble average, allow the molecular dynamics simulation with enough representative conformation to calculate information on macroscopic property using a flexible amount of computer resources. So this says, I mean, if you have this like molecular dynamics, where the constituent of the system are allowed to interact according to the law of physics over a period of time through the numerical solution of the equation of motion, one obtained the trajectories. So we will have the trajectory at the end, and trajectory is nothing but the information about position coordinates and the velocities at particular position of all the constituent of the system under the influence of interacting potential. That is very important. What kind of potential you are applying on the atom, right? So, so these trajectory, whatever trajectory you will get, are then analyzed in order to extract the desired property of the desired property, like as I told, the pressure, stress, diffusion, velocity, surface tension, dielectric constant, other parameters. Since the molecular system generally consists of vast number of particles, it is impossible to find the properties of such complex system analytically. That's why we need computer, the implementation of molecular dynamics simulation with the help of high-end computers. So we cannot do experimentally all this. Like, even though if you have the crystal structure, right, X-ray structure, that is also the snapshot. What will happen after a fraction of second, we do not know, right? We need to, for this, we need to have so many pictures, so many snapshot through the X-ray. That is very, that is not economically viable. So for that, we need some kind of technique, computer techniques through that we can observe the sequential the sequence of, or you can say the consequence of that particular complex in the physiological condition. The MD simulation exercise circumvent this problem by using the numerical solution of the equation of motion. So as you can see here, we have the motion equation of second law of motion by Newton's, and it says that F equals to ma. So here F is the force act on the particle, mi is the particle, and mass a is the acceleration. So for every instant, I mean after a particular time delta t, which I will show the next slide. So every few seconds or every particular time, we need to calculate this f, and based on this f, we need to observe the position of the atom. And after having the position, after having all this position, we will apply some numerical solution, or you can say that we will solve this equation with the help of another kind of function, that is the Verlet function, and that I will show you in the next stage. Next slide, yeah. The potential energy of the function of atom position of all the atoms in the system, because this function is complicated and must be solved numerically with the numerical algorithm, that is Verlet algorithm. So Verlet algorithm says that you need to observe the position and velocity at particular time delta t, and what will be the position at minus delta t. So and these both equation you can see in the third equation, where we are able to calculate the position as well as the acceleration and at particular time, which is delta t. So every time we need to calculate this equation, and all together this will give you the trajectory. I mean, this is the trajectory information. You will have the position as well as acceleration, right? The available potential energy functions such as AMBER, CHARM, GROMOS, OPLS provide reasonable good accuracy with the reasonable good computational efficiency. So for this, you need to have implement the force field for the system, that we'll see in the later stage. What is the force field, why it is required, and which force field is appropriate for the system. So now we will see the main stages of involved in the molecular dynamics simulation. In general, there are 3 stages in the simulation: preparation of the input, production, molecular dynamics simulation, and the analysis. The first stage, it has a multiple stage, include the generating the topology. Here the topology means it contains all kind of information about your input. If the input is protein ligand complex, it contains all kind of information about the complex. What is the chain? How many amino acids are there? What is the you say the atoms? How many atoms are present in the ligand? Everything. Defining the box, we need to define a box and fill it with the solvent if it is explicit solvent dynamics, right? So, so we need to suppose in this case I'm dealing with the protein ligand complex, for example. So in this case, we need to dip this protein ligand complex into a water box. It contains solvent like H2O2, and after that we need to add some counter ion to neutralize the system. Why we need to neutralize it? Because if the system is charged, we do not able to observe the actual you can say the position and the acceleration facing whatever the atom is facing, all these things like pressure, what is pressure, and what you say the temperature and everything. If the charge is not neutralized, it will also add up. I mean, this the charge effect will also add up into the actual observation. So you need to neutralize the system with the help of either sodium ion or chloride ion based on the charge present in your complex. If it is positive, if your complex or you say if or your input is positive, then you need to neutralize with the negative ions. If it is negative, then you have to neutralize with positive ion. Then after that, energy minimization. You after making this system neutralize, you need to minimize it. So the system gets ready for the further steps. And after that, you have to perform equilibration for sufficient time to get the stable pressure, temperature, and energy. So here the equilibration means the position restraint dynamics, where you will restrain or you can partially control your protein ligand complex with some force, and let the water and the solvent nearby are allowed to enter into the protein and the protein ligand complex. That is called the position resistance dynamics, and in this equilibration generally we perform this. And the second stage is the production, where we are not allowing any restraint to the protein ligand complex. We are just making it free and applying the force for particular time. So longest stage resulting the trajectory containing coordinates and the velocity of the system. So that is, you will get the trajectory of coordinates and the velocity. That generally we are not able to find out analytically or experimentally. And the last is the analysis. Whatever the trajectory we got at last, we need to perform the analysis. Whatever the analysis you want, you can choose, like root mean square deviation, root mean square fluctuation, radius of gyration, solvent accessibility surface area, or principal component analysis, or MMPBSA. Everything you can perform with the help of trajectory. So first step, preparation. How you can prepare the protein ligand complex for your simulation. So either generally we prefer the protein ligand complex should come from the crystal structure. Because why it is like that? Because crystal structure are believed to have exact position of the poles as well as exact 3-dimensional structure of the protein, like in the real time and in reality. So that is the real picture which is coming from the experiment. If you have that structure, that further prediction which you are going to do with the MD will be correct. Or if you do not have this, then we will perform. Then we need to get it. Either first we need to model it, then do docking, and then take the best structure, minimize that structure and all. That also you can start with. So after having this, you need to remove the heteroatoms, water from all water molecules from the PDB, and your PDB should have only heteroatom as a ligand, and the atom is your coordinates of your protein. Once you have downloaded the structure, you can visualize it with the help of different visualizers like VMD, Chimera, PyMol. Once we have look at the molecule, you are going to want to strip out the crystal water and other molecules, because that is not necessary. And topology can only be assembled automatically if an entry for a building block is present in the RTP file. So again, this is you can say the limitation. You should have the RTP. In general, we have all kind of RTP available in the with the software. Generally, in this for this stages, I am talking about the GROMACS, which you can get the detailed tutorial on the given link, which is there in the given by the Lamluk, Dr. Lamluk. And so that you need to since this is not the case, we will prepare our system topology for the two systems. So in general, the GROMACS have topology, or you can say the force field information of all the amino acids present in the protein, as well as all kind of atoms that is present in the ligand, like carbon. Force field information in the GROMACS. But how this combination, suppose you have kind of ligand, suppose X ligand you want to you have the X ligand in the protein, but for that X ligand you do not have, you will be not having the RTP file, or you can see the force field file in the software. So for that you need to generate it by yourself. So protein you can generate it with the help of software itself, but for ligand you need to you need to take the external sources like ProDrug, ProDRG, and other servers. So the first is prepare the protein topology with the first command PDB to GMX, and second one is, if you have the protein ligand complex, generate the ligand topology using the external tool, that is ProDRG is very famous and very frequently used. Preparing the protein topology, when you believe you have a good starting structure, the next step is to prepare it for simulation. Romex can do it automatically for you with the tool of PDB to GMX. You can get the same some help on the available options like PDB to GMX, this flag edge. In this case, we'll have the command like GMX, PDB to GMX, then flag F. F is the input, whatever the input you want to give either in the form of PDB, then output, right? And water SPC, that which model you want to use in this simulation, that is SPC. Apart from that, you can also ignore your hydrogen by flag IGNH. You can imply the IGNH, so you won't get any error in that. After that, include the file topology. You will have the at last, after processing this step, PDB to GMX, you will get the topology of your protein molecule, and the ligand molecule you need to prepare it from another source. Preparing the protein topology, the program will ask you for a force field. So PDB to GMX will ask you a force field option. Which type of force field you want to use? The force field refers to the function, form, and parameter set used to calculate the potential energy of a system of atoms or coarse-grained particles in molecular mechanics, molecular dynamics, or Monte Carlo simulation. The parameters for a chosen energy may be derived from experiments in physics, chemistry, and calculation in quantum mechanics. So the basically force field is the selection through which you can define your simulation. I mean, the force field criteria is very important, and we have the almost 18 to 20 force field in the present GROMEX version, and what kind of simulation or what kind of observation you want to know with the help of simulation. Based on that, you need to select the force field. In general, protein ligand complex, the GROMOS, you can see the option number 9 is more frequently used. That is 43A1 force field. And because it contains why I'm saying this option, because the external source like ProDRG, whatever you are using, that implement only this kind of force field. You do not have several options in the ProDRG. So if you are getting the ligand from the ProDRG and ITP files from the ProDRG and mixing it with the different force field, definitely you will have the wrong answer at that time. The wrong observation you will get. So the GROMOS 96 option number 9 is most appropriate while selecting the force field. The force field appeared in the 1960s with the development of molecular mechanics method, and their primary goal was to predict the molecular structure, vibration spectra, and the enthalpies of isolated molecules. Since then, the scope of research has moved to deal with such more complex systems, leading to the development of more widely applicable force fields. The good example are the Dreding and the universal force field that contain parameter of all the atoms in the periodic table. So this is one of the force field which has all the parameters. Whatever the periodic tables has elements, all elements is there in this. Other very popular is Charm, Amber, Gobos, OPLS, and Capacitors. Each force field has its particular strengths and weaknesses related to the data and procedure employed in this parameterization. So the final choice depends on the particular problem being considered. So based on what kind of problem you are handling, based on that you need to select the force field. In general, very basically, very primarily, your force field should contain all the parameters, whatever parameters are required for your structure, like ligand structure or protein structure. If you are having DNA or RNA, so then this Gromos is not a good choice, right? So at that time, you need to have OPLS or CHARM. CHARM is most recommended because it has all kind of information required for the DNA or RNA simulation. Preparing the ligand topology, as I said, that you need to have an external tool or program or server to prepare the ligand topology. We must now deal with the ligand. Proper treatment of ligands is one of the most challenging tasks in the molecular simulation. Automated tools are greatly preferred. For each force field, there are methodologies or software program that's supposed to give the parameters compatible with various force field. Now, not all of them are equally accurate. So we have some most frequently used is the Gromos 87 or Gromos 96, that is ProDRG, and automated servers are topology generation. And the newer server for topology generation also we have Gromos 97 54A7. Apart from that, you have CHARM GUI for the CHARM, or Amber, we have anti-chamber, and OPLS, we have Topol build and LibRegGen. These are the servers through which you can generate the topology. Preparing the ligand topology. We must save the protein and JZ4 ligand into separate coordinate files, save the coordinates so that we can get the different. We need to differentiate it. While starting itself, you need to remove your ligand, process it through the external server, and your protein through the GROMEX. Copy the contents. These are the steps which you need to perform while preparing the simulation system that you will see in the next second half, how we can do individual step. But before that, you should know what is the topologies and how we can generate, why we are generating it, why the ligand topology we are not able to generate through the GROMEX, why we are doing external, why we are taking help of external source like ProDRG server, because the GROMEX do not have all the parameters for your ligand. That's why we are taking it. After taking the topologies from ProDRG, we need to mix it, right? This file would be used to build the protein ligand complex in subsequent steps. Build the complex. So after processing PDB to GMX, you will be having grow file .grow. In this case, you have I given the name of this because I took this all from one of the tutorials that I told before. So you will be having the grow extension that you can rename it to anything. So PDB to GMX, you will be having a grow file, topology file, and the ITP files. So this grow file is contain the information of your protein, and from other source, the ProDRG, you will be having another grow file that is for your ligand, that is in the form of JZ4 grow. This grow file should be combined in the further steps. So we will combine it and we'll give the name as a complex.grow, right? So whatever the coordinates present in the JZ grow, we'll copy it and we'll paste it at the extreme down, and the same number, suppose we have 32 or 39 or 52, whatever the atoms present in the group file, we'll update it in the complex grow. We'll add the in the protein with the protein coordinates, and we'll add this ligand coordinate also. We'll write it on the top. And if it is wrong, then the software, or you can say the GROMEX script, will not able to read this complex file. If the calculation or use the sum of this ligand and the protein coordinates is wrong, you are not able to get the next, you cannot process the next step. Including the parameters for the JZ4, like how we join the GROMEX .gro file, in the same way we need to join the topology file also. So for the topology, we need to include this entry just before the water. Water topology is there. This is already there in the topology file of the protein. So we need to introduce these two lines, Ligand topology, and this file should always be there in the folder, uh, which we are taking from the proteology. The topology and the coordinate files are now in the agreement with the respect to the content of the system. Now we have the the complete complex. So while doing the complex simulation, you need to process by this way. We have to run PDB to GMX, remove your ligand, get the topology from the external source, join it, join the GRO file as well as the topology file. Adding solvent, water around the protein. So since we want this simulation to be accurate, we want to add the water. We want to add the solvent. However, before adding the solvent, we have to determine how big the simulation box should be. And again, if you are making the simulation box is very big, then your speed will be slow, because you need to simulate entire solution, right? Entire solvent, including your complex. So the simulation box should not be so small as well as should not be so big. So, if it is very small, then your protein complex will touch the boundary and you will get the artifact in your trajectory. So for this situation, you need to have a proper simulation box where you can put your protein ligand complex. What shape to use, that is also important. If your protein is long, very long, like it is not in the spherical or something like that, and if it is has a lot of chains in that, then you need to have a rectangle box. And if it is circle, or you can say kind of, at that time you can choose cubic box. Cubic box is more appropriate most of the cases. There should be at least half or cutoff between the molecule and the box side. That is very important. It should not be so close to the complex. At the same time, it should not be so far from the complex. If it is very far, it will be a complex simulation. Due to the restricted time, for further reduce the volume of the box, we will have the rhombic dodecahedron box. This is one type of the one kind of a shape of a box, generally which is there for the big protein ligand complex. If you have very big, then you can use this type of option. Otherwise, cubic is most appropriate. And D is the size of box and the extreme of your protein ligand complex. Okay, adding and the last step before simulating is to add the water in the box to solvate the protein. This is done by using the small pre-treatment system of water coordinates that is repeated over the box, and overlapping water molecules removed in GROMEX. A suitable command to solvate the box is solvent. So why we are adding solvent? As I told before also, if you want to observe the actual physiological action of a ligand, or if you want to observe the stability of a protein ligand complex, you should keep that complex into the solvent box, because in general the drug molecules interact with the protein surrounded by the cytosolic, or you can say the physiological solvent will be there, pH is there. You'll be having a particular pH of that solution. So everything you can maintain in this solvent box. You cannot run your simulation in vacuum, right? Sometimes you can run the simulation in vacuum also, but to get a real picture, you should have a solvent dipped into your the complex molecule dipped into a solvent box to get a real picture. So we have a command like gmx to solvent. So in CP is the input, which generally already predefined box is there where you have ligand, protein ligand complex. We are providing another row, another force field, that is SPC, which we select in the first command for water, and the topology and the output of the solvent. So this file will be having, the solvent.grow will be having your protein, ligand, and the solvent, and whatever the ions you add before for neutralizing the system. Okay, so now is the adding ions. So after creating the box, you need to add ions to neutralize your system. So this for this you need to, again, you have a command like gdion based on your pH of based on your charge of your system. You need to add counter ion to that. If it is negative, you need to add positive Na, or if it is positive, you need to add chloride ion. So based on that, it replaces the solvent, particular solvent molecule, and it occupies the space in the simulation box. After having all these things, like after adding the complex, you need to minimize your system. You need to minimize your system by implementing energy minimization. By adding hydrogen, whatever the things, why we need to minimize the system? Because after adding this ions, after adding this solvent, or whatever you have added into the box, which is required for the physiological condition, that can create some artifact if it is not properly minimized. Energy is not properly minimized, and if so, before starting the simulation, you need to minimize your we need to minimize the complex energy into the simulation box, or entire simulation box you need to implement for energy minimization. So this we can do with the empty run, and before that we had to include one topology file which says all the parameters, like how many stages for minimization, what is where to stop, which algorithm should use for the energy minimization. Either it is steeped descent or conjugate gradient. Which algorithm you need to apply, or both you can apply here. So this everything there in the MDP file that you can see while you are practicing the GOMEX session. The equilibration of the water around the protein. So this is very important stage. After minimizing this simulation, you need to establish the equilibration in the simulation box. So equilibration is also called the position resistant dynamics, where you are running your small dynamics run like 10 ns or 20 ns based on your requirement. In that you are applying the constant, or you can say the restraint to your protein ligand complex, and other molecules or water molecules are unrestricted. So what happens? So at this stage, the water molecules and other things can soak into the protein ligand complex properly before going to the simulation. And if you are not performing this, then it will act like a like all oil balls into the you can say the, if an iron ball, not iron ball, if you take an example of cotton ball surrounded by the oil, right? At that time solvent cannot enter into that. So for applying, to get a real picture, your solvent should be properly soaked by the your protein ligand complex. So that's why we need to apply this equilibration, and based on different ensemble like NVT, MPT simulation, isobaric, isochloric, we need to perform this simulation for particular time duration. So in this is the NVT NPT simulation for particular time. At the time, whatever the water molecule is there, or solvent is there nearby, surrounding by the in the box, it will be properly soaked. Okay. after that, we need to run this simulation for a particular time. This dynamics is unrestrained. I mean, there will be no restraint. We are not applying any restraint to the protein ligand complex. So this process just like to have before, but we do not have restraint in this. D spore is one of the criteria where we are implementing in the MDP file, but in this MD file, we do not have that particular criteria. D spore you will not get. So the D spore says the restraint, but in this we are not applying any restraint to the protein ligand complex. Running the production run. So in this way, equilibration production run is minimal. Upon completion of two equilibration phase, the system is now well equilibrated at the desired temperature, pressure, and we are not ready to release the position restraint and production run. So after that, after having the trajectory from this production run, we can go for the analysis. So you will not have to perform all this analysis, but pick one of the couple of them. Like RMSD is most basic analysis, like RMSF, RG. One of the most important fundamental property to analyze whether the protein is stable and close to the experimental structure. That is through RMSD analysis we can check. So you need to plot the output file, that is your XTC. You need to plot it with the help of Grace software, and you will get the expansion, or you can say the behavior of your trajectory. If it is fluctuating lot, then the system is not equilibrated. You need to simulate more in the production run, or you need to do something with your equilibration run also. And other quantity also you can find out what is the interaction that we can see in the thermodynamic binding free energy. Like if you want to observe the actual binding energy between ligand and protein, then you need to perform this GMM PBSA. This is the tool through which you can get the different energies, binding energy of between protein and ligand, and for the entire trajectory. And based on this energy, you can conclude that particular ligand is which particular chemical molecule could act as a hit, right? Okay, so this is another technique through which you can analyze the protein ligand complex. This is Stair molecular dynamics. So in this, what you will do, that you will pull your ligand molecule away from your protein, and how much force is taken for how much force is required to pull, that will be measured. So if your protein ligand complex is very good and very tight, then you need to apply lot of force, and if it is very loose, then immediately you can remove it by applying the minimal force. So the Stair molecular dynamics is also one of the very good criteria, but it is very computationally very expensive. You need to run lot of simulation in this. So, but you will get the exact result if you implement both MMPVSN and Stair together. Apart from this, so this is the entire MD simulation. So I will give some of the example where we implement this MD simulation and got some potential lead. So this is one of the work which we have done to identify, screen and evaluation of benzosouvinine bioactive molecules against the PI3K receptor. And in this, we have done all this technique like screen the molecule we identify the PI3K by, from starting we had this 17 analogous, that is benzosouvinine molecule, that is ligand, you can say the ligand based screening, and we screen this ligand and we found that PI3K is has more affinity toward this ligand, so screening, and then after we implement the all Edimati and Topcat criteria to check the efficacy of potential leads, and after that whatever the leads we got, we tested into the zebrafish model, and we reported one lead has the best affinity toward the PI3K receptor. Okay, in the second one, this is also kind of the same way we have done that amelioration of cognitive deficit of zebrafish by an undescribed anthraquinone from Jugulans riga. This is one of the plant which is there in the Himalaya, and we got some bioactive molecules. We tested it and we found the binding position, binding mode of this molecule with the help of computational technique, and tested the molecule into in the wet lab, and we found that it has very good activity compared to the standard one. Okay, so this is another work which we have done for the natural bioactive molecule. In that, we had this hesperidine molecule, and we found that this is very affinity. I mean, it has very good affinity toward the BDNF, crab BDNF, and this we proved with the help of wet lab technique. So in this work, we do not perform any wet lab, but we given support to the chemist. So in this work, what we have done, we have some molecules which is semi-synthetically derived from the chemistry lab. The backbone is coming from Citrus Devdara tree, which is widely available in the Himalayas. And this backbone, they created a lot of derivatives, and we found out the most important, most appropriate receptor for this particular molecules, all molecules. And we found that it has very good affinity toward the PPR2 gamma, which is there, which has an implication in diabetes 2. And now we are also testing all these molecules in the wet lab on rats, animal model, rats, and this rabbits. We're testing all these molecules. Either they have real affinity, or they are not they are really changing the disease condition or not. So this is another work which we have done. This is completely computational work which we have done on identification of the bioactive molecule of Pcna. So in this we took a co-crystal structure which has a very good affinity toward the receptor, and we screened the entire database, whatever we have here in the ISBT. We screened it, and we found this two potential molecule having very good affinity, and after that we tested it with the help of molecular dynamics simulation. Apart from this, in the COVID scenario, we screened almost 6 to 7 receptor of COVID, SARS-CoV-2, and we found out bioactive molecules for each of them, and we published each of them in very reputed journal. In this all, we have done computational tasks, molecular dynamic simulation, MMP, BSA is shared, everything we have done in this. So like NSP1 to 16, spike, RNA dependent alanine polymerase, main protease, we have screened for everything, all these receptors. And the main protease in the spike, we are going for the wet lab, and we got whatever predicted molecule. We are getting very good results from them. And after that, we tested these predicted molecule into against the live virus, and we are also there also we are getting very good result. So as you saw that we are implementing this molecular dynamic simulation. So it is not a new way to discover the new lead successfully, because there is a lot of FDA approved drugs coming from. I mean, all the drugs are coming from rigorous molecular dynamic simulation and CAT techniques. So in this slide, you can see the emprinavir, grazinovir, retrigranavir. This is the HIV integrase inhibitors. This particular two inhibitor like emprinavir and retinavir, this basically it is came into the light after MD simulation. They have done very long simulation against HIV protease, and this in this case HIV intrigase, and they reported this is that could be a potential. And later stages, it proved that this drug is very potential or acting as a very good against the HIV. And later the FDA approved these molecules against so many drugs which is coming which is there in the list of FDA approved after after the MD simulation or computational aided drug design efforts. So this is our lab in ISBT Palampur, and we are doing the structural bioinformatics task where we are screening the molecule against the suitable targets. If we have the molecules, we are performing ligand-based screening, and if we have particular disease, or if we are want to design some molecule, we are doing the structure-based screening also. And after screening all this molecule, we are generally we test those molecule into in vivo evolution and cell lines. So this is the entire flow of our laboratory. At last, this is my group. We have 4 students, Mr. Sachin, Ankita, and Vijay and Rahul. These 4 are here currently, and one is already passed out from this lab. Thank you very much.
Thank you, sir. Now I would like to ask from the participants, if you have any questions, you can ask Dr. Rituraj Purohit. Or there are 2 questions in the chat box from Pujit and Rajendran. How missing residues affect molecular dynamics?
Very much. If the missing residues are there, you will get the trajectory, or you get the structure break during the dynamics. That is very important issue which you need to take care. Most of the PDB structure, not most of the, you can say 60% PDB structures have a missing residue. So before that, you need to model it. You need to you can check those residues. I mean, you can check this missing residue through Chimera, and if you find that it is there, you need to model it before starting the MD simulation.
Okay, so the next question is a very simple one. Do we need a specific system requirements, mean computer and processor, to perform molecular dynamics study?
Yes, you need to have very specific system which have very good amount of RAM and the processing power. If you do not have, your system will get hang, or subsequently it will get crash. So in general, in our lab, we have the HPCs, high-end HPCs, which has very good amount of RAM, and even we have the GPUs also, through which all the big simulation we used to run on the GPU.
Next question is from Richa. How simulation helps in defining 3D structure of a protein?
Okay, so for 3D structure, define the 3D structure of a protein. Suppose if you model the protein, right? And after getting the model structure, you need to validate it. If it's perfectly fine, you can simulate it to identify the correct orientation of all the loops present in the structure, side chains of the structure. So for refining the structure, this simulation is important. Okay, I can read the another one. How can calculate the inhibition cos Ki in the molecular docking? You can do that if you observe the log file in the molecular docking. If you are doing the autodock, you will be having the molecular log file, right? So in that log file, you can see the Ki value. Just Control F and find out the Ki. You will get the Ki value of each pose, right? Each ligand pose or each ligand. Sir, can we repeat? Can you repeat the information regarding how to choose force field for the simulation? Okay, first selecting the force field, you need to have proper information about your system. If you have the DNA or RNA into your system, then you have to choose OPLS, right? OPLS or CHARM. Generally, CHARM will be more preferable if you have the DNA or RNA. Or if you have general protein ligand complex, then you can go for the option number 9, that is Gromos 96. That will be most important, I mean, recommended, because the ligand topology you are getting from the ProDRG, that server used this Gromos 96, and it is compared with the Gromos 96 of the Gromex. So you should use the Gromos 96 while processing the protein ligand complex. How to do energy minimization, energy calculation using Gromos software. So energy calculation, for that you need to use the GMMPBSA script, which is there in the web. You can, after getting the trajectory, you can you can analyze your trajectory with the GMMPBSA. You will be having free energy calculation. You will be having free energy of your complex. How we can determine minimum time required to perform MD for new complex. This is somewhat confusing, Anjali. I'm not getting your question. Okay, next, Pujit. Can protein in a water simulation help us to find the protein misfolding due to SNPs? Yeah, definitely. This is a very general practice. All the SNPs previously in long back during my PhD, our group used to do that. Whatever the SNPs or what are the mutation information, non-SNPs, we used to induce into the protein, and we used to observe that particular effect of that SNPs into the through the help of through molecular dynamic simulation. Okay, Iftikhar Ahmad, please tell me the name of free tools available for study QSAR. QSAR, I think QSAR already covered by Dr. Firoz. I hope he has discussed all this. Any more question?
So participants, do you have any more questions to ask on molecular dynamic simulation from Dr. Rituraj? So, sir, there are no more questions in the chat box. One question comes: how many nanosecond is a must for a simulation?
Okay, so this is very most frequently asked question. How many nanosecond, picosecond, and all. So time is based on your system size. If you are running a peptide, right, small or near about 50 or 75 amino acids, then 100 or 200 nanosecond is perfectly fine. But if you are running a very big complex like Pcna, which I talked here, it has a lot of chain or multi-mer complex. At that time, you need to have a simulation for very long duration, and because the system is very vast, and to observe each of the atom, you need to have a big trajectory. So based on your simulation, I mean the system size, you need to decide the simulation time. And moreover, you can see the RMSD also. I mean, if the RMSD is converged perfectly, right, then you can stop the simulation. If the RMSD is fluctuating very much, then you need to do more simulation for longer time.
Okay, so are there any more questions? I cannot see any more questions in the chat box. If anybody is interested, you can ask a question, a very quick question. So I think Dr. Rituraj Purohit, there are no more questions to be asked. So
Yeah,
I thank you on behalf of the organizing committee and our participants. You gave a very good insight into molecular dynamic simulation, because people have, even my students have several doubts about it. So it was a really nice and informative talk. I would like to thank you again for coming and giving us such a informative lecture. Thank you so much.
Thank you very much, ma'am, for inviting me to my the alma mater, and It is always pleasure to talk with the student, or whatever the student doing in the current field. So thank you very much.
Thank you, sir. So, dear participants, we are at the end of the second talk of today's schedule. So, we'll meet at 2:30 p.m. sharp for practical session on docking and molecular dynamic simulation. So, kindly join 5 minutes before.